So we were looking at some of the factors that affect the toxicity of a chemical, and I'd like to take a minute more to review some of these in more detail. The first one is bioaccumulation, and you'll remember this from several chapters ago. But bioaccumulation is when there's an increase in the concentration of a chemical within an organism over time. That particularly happens when a particular chemical can be dissolved in fat, so stored in fat, so it accumulates. The second one was biomagnification, which is when there's an increase in the chemical concentration in animal tissues as the chemical moves up the food chain. And the good example here was DDT, which is both a bioaccumulator and a biomagnifier. And here's how it biomagnifies as it moves up through each of the trophic levels. And the third factor, persistence, how long the chemical stays in the environment. So some chemicals, such as DDT, have a very long half-life. So let's look at that on the next slide. Okay, this is a chart of persistence of various chemicals. And the first one on the list here is malathion insecticide. That's very popular for fruit trees, so down in Florida. It has a half-life of just 1 day, so that's considered a very short half-life, so it won't persist as much in the environment and therefore is a lower risk. Moving down the line here, you might also be familiar with Roundup. Roundup is an herbicide that's used to control what are considered pest plants. Roundup has an intermediate persistence. It can be as short as 7 days to as long as 70 days in water. So that's a more intermediate persistence, but it's still considerable. When you move up to things like PCBs and DDT, which was shown in the last slide, that can be available in the soil or in water 8 to 15 years, and for DDT in the soil 30 years. Some other defoliants like Agent Orange that were applied in Vietnam in the 1970s also have a very, very long half-life of about 30 years. And remember, it works just like with the radioactivity. So after 30 years, half of it's gone, but that doesn't mean that in 60 years it's all gone. Okay, it just means then you'll have half again as much. So that certainly adds to the toxicity of a chemical. Okay, so now we've talked about toxicity of a chemical and measuring it through lethal dose studies and effective dose studies. And now we're going to look at a different type of study called an epidemiological study. Epidemiological studies are studies on large populations of humans or animals. It can be used to study both chemicals and biological agents. So, epidemiology is defined as a field of science that seeks to study causes and patterns of disease in animal and human populations. And these can be tricky to do. There are several types. The first type is a retrospective study, which monitors populations that were exposed to a chemical at some time in the past and looks for possible effects. So I have an example for you of a retrospective study, and this is also an example of environmental justice or injustice. So in the mid-1980s in Bhopal, India, there was a fertilizer plant that was owned by an American company called Union Carbide. And one night, in the middle of the night, they had a nerve gas release. And this nerve gas release, the workers tried to stop, but there were a series of equipment failures so they were not able to prevent the gas from being leaked from the plant in massive amounts. So 2,000 people died that night as a result from exposure, and 15,000 later died from this exposure, for a total of 17,000 cases. So this population that was exposed to the nerve gas was monitored for a couple of decades and compared to people who were not exposed to the nerve gas. And they were found that to have higher respiratory symptoms, higher stillbirths, higher rates of genetic abnormalities, infant mortality, kidney failure, and learning disabilities. I have attached a video clip to Moodle that will tell you more about this incident from an environmental justice point of view. Okay, a second type of epidemiological study is a prospective study. And you start with a group of people, hopefully a large group of people, and monitor that population and have them record things like foods that they eat or alcohol that they drink over a period of time, typically decades, to determine if certain illnesses occur more frequently based on what they had been exposed to. So a typical example of that is the nurses study that was started many years ago. And in the nurses study, they asked the nurses to record what they ate every day and submit that data for a year. And then after that, they had them continue to record their data, but going back and saying, what did you eat for the past month? And eventually to recording things like, what have you eaten for the last year? A couple of problems with those types of studies is, for one thing, the nurse might have recorded what she thinks she should have eaten. So maybe she remembers to put down that salad she has for lunch, but didn't record that lemon meringue pie that she had for dessert last night. The other problem is that it was okay when they were doing it on a daily basis, but to expect people to accurately recall what they have eaten for the past month, much less the past year, often doesn't happen. And the last problem with this type of study is that there are so many different foods that we might be exposed to that it's really hard to target even a category such as exposure to a certain amount of calories or exposure to sugar or exposure to a certain amount of fat. So these studies too have their issues, but they might shed light on some things. The best type of study is a prospective, randomized, double-blind study. And that's not really discussed in your book, but what that means is a group of people are chosen, half are put into one group that will actually receive the test chemical or medication, and half are put into another group where they will not. They'll receive a placebo. And it's randomized. They're just assigned to these groups, not based on anything other than a mathematical system. And the double blind refers to the fact that the first blind is the patient doesn't know whether they're taking a chemical or a medicine. And the second blind is that the doctor or nurse administering the medication doesn't know whether that medication is the placebo or the real McCoy. Because if they knew, they might have some bias and the data might not be quite as accurate. Okay, so this is a good time to take a look again at the roots of exposures from both bacterial, biological diseases and chemical illnesses, toxicities. So let's look at the roots of exposure and compare them. So for infectious disease, we had the domesticated and wild animals. We had human to human spread, food to human spread, air and water to human spread. And one thing that your book did not have listed in this diagram that is important is that an infectious disease can also be passed from the mother to the fetus or the baby through breast milk or from, delivery and everything else. So chemicals, very similar. Chemicals can come from air and water and food, just like they can for infectious diseases. Also can come from soil. And chemicals, again, can be passed from the mother to the fetus and baby. Okay, now we move on to the last major topic, which is risk analysis. So risk analysis is a process that we go through to determine whether a particular item, be it a chemical or a biological illness, what is that risk to us? What level of risk can we accept, and how can we manage the risk? So let's start here at risk assessment. Risk assessment includes identifying the hazard, characterizing its toxicity, in other words, do those initial studies such as the dose response studies, and determine the extent of exposure. Then you move to risk acceptance, determine the acceptable level of risk balanced against social, economic, and political considerations. And then risk management, determine the policy from private citizens, industry, and government and interest groups. So even though here we're primarily talking about chemical risk, I think it would be worthwhile to look at it in terms of COVID-19 risk and how that has progressed over recent months. So first was identifying the hazard. As you know, it was first identified in Wuhan, China. And then characterizing the toxicity. In this case, it's characterizing how contagious is it? How much damage does this do to particular populations? And we're still working on that. We don't really have a full handle on the toxicity of COVID-19. And then determine the extent of exposure. Who has been exposed already? Who is most at risk to be exposed? Then moving over to risk acceptance, we have to balance social, economic, and political considerations, and boy, have we ever seen that with this disease, right? So we have social distancing now, and that's been very hard for a number of individuals, including, I'm sure, you and I. Then there are economic results to that. The economy has just come to a screeching halt, unemployment has gone up, stock market down, and then last, there are political considerations. Do we have leadership at the state, local, and federal level? Leadership within our own businesses and institutions such as schools. And that all goes into how much risk we are willing to accept compared with the risk of the hazard. And the last step, which hopefully we are moving to now, is risk management. Determining the policy. When can we lighten up the tight lockdown here? And that's going to come from a series of private citizens, industry leaders, government, and other interest groups, things like the CDC, hospitals, etc. So that's a process that we're in the midst of going through right now. So I'm going to wrap up the screencast here with the risk analysis, and then we'll be moving into perceived versus actual risk as the final topic for this chapter.